Okay, so we'll start the session. Just a minute. Okay, so this is the last class. Uh, Probably the last class. I don't know. You might have one extra class next Tuesday, just for some doubt clearing sessions. So that Tuesday class you might have or might not have. It depends. Uh, most chances are there more chances are there that there will be one more class next this thing Tuesday, just for final doubt clearing session kind of a class. So today we are going to just kind of sum up everything that we have learned in this module. So the module is about genetic engineering. So in this module we have right from the starting of picking up the genomic DNA, the genomic gene of interest from the whole genome, whether you know the sequence or you don't know the sequence. Just a minute. Yeah. After that, cloning of that particular gene of interest into your desired vector. Then we have dealt with the expression of the vector, the expression of the DNA. After expressing it, how we can purify it, how we can characterize it, what are the properties that we can study to know about that product that we have made, and then the ultimately the applications of biotechnology in several fields. So this week class was just about revising of what all we have learned in this module, and quickly we'll go through the not we will not go through each and every topic from each and every class because that would take a lot of time. We'll just quickly see from the presentation that was given to you by sir, that what are the topics that are important and what we have studied, and then we can probably just solve a few questions. And so in the next class, that would be the last class, hopefully on next Tuesday same timing. um, we can think of certain topics that are difficult for you or that you think you need a little bit of revision. Those topics we can take up, and some questions related to those topics, or in general many questions that we can solve. And this week assignment which is given to you, so as you must have already been told in the lectures that the kind of assignment that you have, uh, that would be like the pattern of your final examination. So it won't be like a very difficult exam, I guess. You just need to have the concepts and the basics correct, which if you have really read the classes, I think that would be enough. The notes and whatever information that you have read from the lectures, I think that would be enough for your exam. So anyway, we'll start. So we have studied right from module 1. We have started with the basics of host, that is the basics of the prokaryotic and eukaryotic cells, the fractionation. Then we have started with, then we went to the metabolism. So I'll just do one thing. Instead of going with this, we'll go with the PPT. That would be better. Yeah. So the course module is about creating a genetically modified organism or cloning a particular gene of interest into your desired vector. So for that, we need the gene of interest and we need the vector. Then you have to cut your gene and the vector with the same set of restriction enzymes, or same restriction enzyme if you're doing single digestion. So if I'm using eco R1 to cut my DNA of interest, gene of interest, then I have to use eco R1 only to cut my vector or the plasmid. So for the digestion, you can use either one enzyme or you can also use two enzymes. That means either you can have a single digestion or you can do the double digestion also. So when you are doing cloning, double digestion is what is actually preferred, because the chances of self ligation is also less, and the chances of misorientation of your gene of interest is also less when you're doing double digestion. So misorientation means that when you are doing double digestion and when this gene of interest is going inside the plasmid, the orientation, that means the 5 prime to 3 prime orientation, is can be easily monitored. Okay, so double digestion is mostly preferred. And then after your digestion, you have to ligate the gene of interest with your vector, then you will transform it, and then you will screen it, and then you will get the clone. So this is like a basic strategy, a basic general outline of how to do a basic cloning. Okay, now depending upon whether you just want to clone or you want to express, the kind of vector that you are using, the kind of plasmid that you are doing using, that would differ, right? So if you just have to clone, you can just have a basic cloning plasmid, any plasmid depending upon the size of your insert. But if you also want to express your clone, that means you have to have some extra other sites in your plasmid. So what are the other extra things that has to be present when it has to be an expression vector? What do we need? Anybody, if you remember, if you want your cloning vector to be if you want your clone to express, then what are the extra things that are needed?
Origin of replication.
Origin of replication is basic. So origin of replication has to be there, whether it's a cloning plasmid. Origin of replication is a basic need of any plasmid. You have to have origin of replication. You have to have some cloning sites, maybe multiple cloning site. You have to have some antibiotic resistance. These are the things that are basic, that has to be there in any vector if you want to use any plasmid as a vector. But if you want to use that particular vector, cloning vector, as an expression vector, so what is the difference between a cloning vector and an expression vector? So you have cloning vector and you have expression vector. Okay, so expression vector means that whatever clone, whatever gene that you have inserted, now it will express. That means now here in cloning vector, the basic requirements are your origin of replication, your selection marker gene. It I mean, I have written here antibiotic, but it can be any kind of selection marker gene, and your cloning sites, multiple cloning sites. But when you want to express that gene, that means you want that it should produce an mRNA, then it should form a protein, then you should have what is needed, translation. So translation site, start site, ribosome signals. So these are the things, translation termination sites. So these are the things which are needed in an expression vector, because you want to express it. That means you now, that particular gene of interest will undergo translation. Okay, that gene of interest will undergo transcription and translation. So things that are needed for translation, the start site, the end site, the ribosome binding sites, these things are now needed in an expression vector. So cloning vector will just clone your gene of interest, but expression vector will lead to the production of proteins also. So whenever we are like here, we have studied about downstream processing, and we have studied about uh, I mean, the product characterization, the product purification, how we can purify our protein, how we can characterize it, how we can extract it. So all these things comes when you are using an expression vector only. If you are using a cloning vector, then there is no protein production. So expression vector means that you are expressing your gene. That means there is protein production. Okay. So origin of replication has to be there in cloning vector also. But an expression vector, apart from origin of replication, you have to have start site, end site, with a translation, start site, end site, ribosome binding sequences. These things should be there. Then, so now this is like a general protocol, general outline of creating a genetically modified organism, or creating a genetically modified organism would be a big word, just a basic cloning protocol. So now we have host. in which you put your plasmids, okay, your recombinant plasmids, as well as your non-recombinant and transformants plasmid. Everything is in your host. So now what kind of host you can use? You can have prokaryotic host and eukaryotic hosts, okay. So then we have, in the first module we have studied that what kind of host we can have. We can have prokaryotic host and we can have eukaryotic host. In that we have studied about the basic structure of prokaryotes and eukaryotes, what are the differences between prokaryotic and eukaryotic cells, and in eukaryotes also you can have plant and animal cells. So what are the differences between a plant host and an animal host, and then how you can isolate the different organelles of eukaryotic cells, how you can fractionate it. So these are like very basics. You all have, I think we all are reading this since uh, ninth. So this is like basic, what are the structure, what are the basic organelles, what are the differences between prokaryotes and eukaryotes, just to have an idea of what kind of host variety we can use, okay. So first thing, in we have started our discussion in this particular course is about hosts, okay. So first thing that we have read in our this module is about hosts, whether it can be prokaryote, eukaryote, its characteristics, its differences, its fractionation, everything about host we have studied. Then we started, this is like just basic, it's like a revision of the slides that we have, that what are the applications or what are the areas in which your biotechnology is used. So basic principle of fermentation and baking that we are using since many years, fermentation, then some important milestones, these are all basics. Then it comes the basic organelles and the structures, the plasma membrane structure, the plasma membrane structure of the prokaryotes and eukaryotes, and then about gram staining, that how you can differentiate between gram positive and gram negative bacteria with the help of gram staining. So all the things that you see in your module 1 is about host and host properties and host characteristics, okay. So when you're doing a genetically when you are studying about cloning and genetically modified organisms making, you need to have an information about what kind of host you have to use. So this is all about the host information. Then you have eukaryotic cells, you have animal cells and plant cells, plasma membrane. Then what are the functions of plasma membrane? So plasma membranes are used for, so plasma membrane has proteins, and those proteins can be used as transporters, receptors, enzymes, or they can be used to transduce signals in various metabolic pathways. Then you have the functions like protection, communication, respond to environment recognition, basic principles, okay. Then then we have studied that this is, up till here it is all module 1, right? this is all module this is all module 1. So we have studied the fractionation. That means you have so many different organelles in a eukaryotic cell. So how you can, just a minute. Yeah. So how you can extract the different organelles. So first, sorry. Yeah. So first module was about host. This is module 1. Okay. Then second, we have studied about the metabolisms. Metabolism means that if I am doing some kind of cloning and I want to use a host, only not only that, I want to know that whether the host is prokaryotic or eukaryotic. Why it is important to know whether my host is prokaryotic or eukaryotic? Can anybody answer? What difference would it make if I use a bacteria or any other eukaryotic host? What difference it would make? Both will both will replicate my plasmid, my recombinant plasmid. Then why I need to have the information that whether my host is prokaryotic or whether my host is eukaryotic? Can you think of any answer why you need to have the information, prior information, before starting of your cloning experiments? Anybody who want to try? Why you need to know whether your host is a prokaryote or a eukaryote host? Okay. So you need to know why whether your host is a prokaryotic host or a you. Just a minute. Okay. Yeah. So you need to know about the properties and about your host, because uh, suppose if I want to clone a gene and then I want to express it, so every gene and every plasmid will behaves in a different way. Okay, every different plasmid will behave in a different way. Every gene will have different behavior. So I should have the information that whether my host is compatible for proper propagation of my gene of interest or not. Okay, now what I mean is that I'm in my lab, I'm trying to generate a cell line. Okay, so for that what I'm doing, that I did a cloning, and then I have to transfect my gene and the plasmid, that means my recombinant DNA, into one particular cells. Okay, those are B cells, so I have to transfect my recombinant DNA into the cells. So I'm doing it past 6 months, but I'm not getting the results. So now only I got to know that the problem was my host, that my gene was my recombinant DNA. It was somehow it was producing some toxic substances, and because of the production of those toxic substances, it was not able to replicate, and therefore I'm not able to get my recombinant DNA inserted into the this thing, my cells. So you need to have prior knowledge that whether your gene of interest, when it is getting cloned in a vector, it is producing some kind of toxic environment for the host. So therefore the host and the recombinant DNA should have a compatibility with each other. So you cannot have 100% assurance whether your gene of interest will have, or whether your recombinant DNA will have compatibility with the host or not. So for that you have to do literature search, that whether someone has used this plasmid in any host, and all these studies you have to do. And then you have to see that whether the recombinant DNA that you have produced, it is toxin free or not. So for that you have to do some basic purification steps that will render your recombinant DNA toxins free. So as a whole, the gist is that the knowledge of host, whether it is prokaryotic or a eukaryotic, is important, so that at later stages there are no problems. You should, your host should be compatible. It should have those metabolism or machinery to support the growth of your recombinant DNA. So that's why in the in the first module we have studied about hosts. Then after getting the information of what kind of hosts are there, then we switch to what is the metabolism. Okay, that how those hosts behave. So in the second module we have started with the metabolism metabolism of different hosts. So when we talk about metabolism, so that means that we are talking about the basic pathways that are required for the generation of energy. Okay, it is glycolysis, it is Krebs cycle. So these are the basic pathways by which the generation of energy takes place inside the host cell. So we have studied whether your generation of energy is going through an oxygen dependent or an oxygen independent manner. So if it is going in an oxygen independent manner, what are the products produced, how they are produced, what is the mechanism. Then we have studied about yeah, just a minute. Huh. So then we have studied about the glycolysis pathway, then how many ATPs are produced in the glycolysis pathway. So in the second first class itself, we have had a very elaborate, detailed discussion about how this ATP is getting generated. Then we have studied about that after this glycolysis pathway, how through Krebs cycle, then again the complete generation of ATP takes place. Then if anaerobic oxidation takes place, that means if oxygen is not present, then how the formation of lactate and ethanol takes place, and what are the significances of those anaerobic oxidations and everything. So that means we have studied about the metabolism of host, how the host is behaving, and that behavior we have studied with the help of glycolysis pathway, Krebs cycle. And then if you are going through anaerobic oxidation, that means without the presence of oxygen, then how the synthesis of lactate and ethanol takes place, and what are the enzymes which are required. This is what we have studied in the second module, I suppose. Yeah. So now we'll just stop for a minute and we'll try to solve two or three questions, and then we'll again come to uh the revision part. So the questions today are like mixed, okay? They are not from a particular module. They are just mixed questions, just like you will have in your final examinations. They are mixed questions. So the first question is easy. Non-membrane bound organelle is rough endoplasmic reticulum, smooth endoplasmic reticulum, ribosome, or Golgi apparatus. So, which amongst the following is a non-membrane bound organelle?
Ribosome.
Ribosome. Correct answer. Then, mammalian culture media is sterilized by autoclaving, filtering, dry heat, UV irradiation.
Filtering.
Filtering. So filtering through zero point 0.2 micron membrane. So always remember, so we make media for bacterial cell culture also, for plants also. I don't really have much knowledge about the plant cell culture media because I am a animal person. So but when you are doing any kind of animal cell culture or mammalian cell culture, you cannot autoclave the media, okay? And you cannot just put it for UV radiation, because you what will happen if you're autoclaving? Can anybody tell me what will happen if you put so much of heat to your media? What will happen, whether it is a dry heat or wet heat through autoclaving? What do you think would happen if you are putting so much of heat sterilization for your mammalian cell culture media? Any guess? Any guesses what might happen?
Ma'am, the nutritional value of the contents will be compromised.
Yes. So the nutritional value would be compromised. So why it would be compromised? Because when you're talking about an animal cell culture media, mammalian cell culture media, so you add serum, FBS, mostly fetal bovine serum. So this serum has a lot of things such as growth factors, okay, and other some other nutritional things, hormones, and a lot of things are there in this FBS. So and some also some immunogenic molecules are also there, like complement proteins are there. A lot of things are there this in this FBS. So this is a serum. So serum will help in the proper growth, and it will provide the proper nutrition for the media. Okay, so when you're making a media, you have your media. Your media would be either in powder form or it would be in liquid form. Okay, so powder form is in, like, you have media in a sachet, and you have to take the required amount of water, sterilized water, milq sterilized autoclaved milq water, and then you have to add that sachet in it, and then you have to add FBS in it, and some one or more things depending upon what are the constituents of the media provided, and then you have to just filter it. So that is like a powder form media. In a liquid form media, you have to do nothing. It is already in liquid form. You don't have to mix anything. You just have to add FBS, and then you have to add some antibiotics, whatever antibiotics you want to add, and then you have to filter it. So what happens, that if you are giving heat sterilization to your media, your so nutritional value will come down because FBS would get degraded. So how the nutritional value of a mammalian cell culture media is getting degraded? Because FBS will get destructive. It will get degraded because so many growth factors, so many other things are there, and because of the heat, it will get degraded. So that is why you cannot have heat sterilization of mammalian cell culture media. Therefore, you use filter sterilization with a zero point 0.2 micron filter. And also, one important thing which you should know, that when you are using FBS, so depending upon the kind of usage and depending upon the kind of work that you are using, you can either this FBS, you can either I think you have also read it in the this thing, I think sir has also uh, uh, taught that you can heat inactivate it. So inactivation means that there are so many immune molecules which are also present in the FBS, okay, such as some kinds of antibodies, some kinds of complement proteins. You only want growth factors and other nutritional setup to be present in the in FBS, but you do not want any kind of immune molecules that can hamper with the growth. So when you are heat inactivating your FBS, that means you are putting it at approximately 51 degrees Celsius for 30 minutes in your water bath. You are degrading the immune molecules such as complement molecules and antibodies. So that kind of heat inactivation you do first, and then that heat inactivated media you used for your mammalian cell culture. Whereas when you are doing any kind of bacterial cell culture, so then what you use, like you just you don't use serum, okay, you you just use your LB, and then you just add your water, and you don't really have any kind of nutritional requirement. You The nutritional requirements are supported by LB only, so there is no need of adding FBS. So therefore that media can be autoclave, there is no problem. But when you're doing a mammalian culture media, it cannot be autoclave because FBS will get degraded. The next question is, how many DNA duplexes are obtained from one DNA duplex after 4 cycles of PCR? These questions we have already studied. Okay, I have just kind of compiled some questions, and some questions are new, some questions we have already dealt. So, how many DNA duplexes are formed from one DNA duplex after 4 cycles of PCR? What is the formula?
Mom, 16. 2 raised to the power 4.
So, the formula is 2 to the power n, okay, where n is equals to the number of cycles. Number of cycles. So here the number of cycles are 4. So 2 to the power 4, which comes out to 16. Therefore 16 is the answer. So how many DNA duplexes are formed after n number of cycles? That is 2 to the power n. Then next question is, DNA sequence having highest melting temperature is, so which amongst the following has the highest melting temperature from here?
Mom, D.
Okay, why you say D?
Mom,
D is
it's
the
a
correct
GC
answer.
request.
Yeah, so it is a GC rich sequence. So A has two double bonds with T, whereas G form three hydrogen bonds with C. So that means a bond which has GC is difficult to break than a bond which is AT. So therefore, I think we have also discussed when I was taking this class. So whenever you are doing primer designing, it is always recommended to have approximately 55 to 60% of your GC content in your primer. And it is not a rule, but it is better that the last uh, the last two nucleotides, if they are G or C, it's better. Of course, it's not always possible to have a primer which has G or C in last, but it's better if you have G or C at the last. Then, yeah. So Pontinus pyralis is an insect source for PFU polymerase, luciferase, T4 DNA ligase, SF9. What is the answer? So the answer is that it is a luciferase enzyme. Okay. All the pyralysis, they are mostly used in your, just a minute. Okay. So they are mostly used in luciferase assay. Mm, just a minute. Okay, I was getting confused. It is, this is the spelling. 14F. Yeah, sorry. Okay. The next question is the vesicular drafting system comprises of mesosomes, mitochondria, lysosomes, mesosomes, Golgi bodies, endoplasmic reticulum, Golgi bodies, lysosomes, Golgi bodies, mesosomes. What is the answer? What is vesicular drafting? Anybody remembers? Anybody remembers what is vesicular drafting? Okay. What is the difference between rough endoplasmic reticulum and smooth endoplasmic reticulum? Can anybody tell what what is the difference? What are RER? What are rough endoplasmic reticulum? What do they have? Anybody? What does RER stands for? It's very simple. We have been studying it since, I don't know, maybe ninth or 10th grade. RER, SER. Nobody remembers. Nobody remembers what is rough endoplasmic reticulum and smooth endoplasmic reticulum? Okay, fine. So, rough endoplasmic reticulum has ribosomes attached to it, whereas smooth endoplasmic reticulum does not have ribosomes attached to it. Okay. Now, so one thing from here we get to know is that some endoplasmic reticulums have ribosomes attached to it. So, what happens actually is that protein synthesis it takes place in the ribosomes. Okay. So, protein synthesis it occurs in ribosomes. Fine. So in ribosomes when the proteins are synthesized, just a minute. It's okay. So in the ribosomes when your proteins are synthesized, then what happens that these are very naive proteins. Naive proteins means that proper modifications, proper foldings of these proteins has not yet occurred. So for the proper modifications and the proper folding of the proteins, these proteins are then sent to the rough endoplasmic reticulum. So from ribosomes the proteins go to the endoplasmic reticulum. Okay, so in the endoplasmic reticulum what happens, these proteins get some kinds of modifications. What kind of modifications happen in endoplasmic reticulum? Glycosylation, N-linked glycosylation. We all must have studied that in endoplasmic reticulum, N-linked glycosylation occurs, and also a lot of modifications which helps in the proper folding of the protein that also occurs in the endoplasmic reticulum with the help of chaperones and co-chaperones. Okay, so the protein, the naive protein which is synthesized in the ribosome, it moves to the endoplasmic reticulum. In the endoplasmic reticulum, it gets N-linked glycosylation modification and proper folding modifications. And from the endoplasmic reticulum, it then moves to the Golgi bodies. In the Golgi bodies, some other few kinds of modifications which are left, such as O-linked glycosylations, those modifications occurs in the Golgi bodies. And then Golgi body kind of direct it where it has to go. Okay, so the proteins which are made for, let's say, plasma membrane, so the Golgi body is the center which kind of dictates or directs each and every protein where it has to go. It will assign that okay, this protein will go to plasma membrane, this protein will go to mitochondria, this protein will go to some other place. So like this, the Golgi bodies is responsible for that direction where it will go. And some things which are not completely properly made, then they can also be degraded. So then for degradation it will go to lysosomes where it will get degraded also, or lysosomes will also keep it take it to the plasma membrane also for whatever function it is needed. So this is your vesicular trafficking, that with the help of Golgi bodies, this bodies secretes the vesicles, and these vesicles kinds of pinch out from the Golgi bodies and take it to the places, or it can also take the help of lysosomes, and it can, if it is not properly made, it will degrade. If it is made in a proper way, it will just perform its function. So this is your vesicular trafficking. And this, so the answer would be endoplasmic reticulum, Golgi bodies, and lysosomes. Always remember basic that endoplasmic reticulum can be of two types: smooth endoplasmic reticulum and rough endoplasmic reticulum. Smooth endoplasmic reticulum, no ribosomes. Rough endoplasmic reticulum, because it has ribosomes attached to it, therefore it looks like, microscopically it looks rough, therefore it is named as rough endoplasmic reticulum. Rough endoplasmic reticulum is involved in vesicular trafficking and not the smooth endoplasmic reticulum. In smooth endoplasmic reticulum, some kind of detoxification of some metabolic compounds takes place. So smooth endoplasmic reticulum is responsible for detoxification. Okay, next question is, the restriction site cuts only methylated DNA but not unmethylated DNA. So this is an important question. Which restriction enzyme cuts only methylated DNA? Which restriction enzyme cuts? We have studied it. We have solved this question also in one of the classes. Anybody remember?
Ma'am,
Ma'am,
DPN1.
DPN1. Yes. DPN1 is the enzyme which is used for cutting the methylated DNA. So EcoR1, BAMH1, HIN3, these are like very most commonly used enzyme. They are very commonly used in our normal this thing, normal cloning. Then eighth question is, T4 DNA ligase enzyme was isolated from T4.
Virus.
Bacteria.
No. Think
Ma'am,
more.
virus.
Virus. So what are the two types of different DNA ligase? One is bacterial DNA ligase, other one is T4 DNA ligase. So what is the full form of T4 DNA ligase? T4 stands for,
Ma'am, it's the name of a bacteriophage.
yes, so T4 bacteriophage. So T4 stands for the bacteriophage. T4 bacteriophage. So DNA ligase which is isolated from T4 from a bacteria which is infected with T4 bacteriophage. Okay. Yeah. Next question is IP simple. IPTG is a synthetic analog of,
Lactose.
no. Sorry. Lactose. Yeah, you are right. Lactose. Then next question is, SF9 cell line was derived from SF9. Remember SF9?
Ma'am, insect.
Yes, insects. Okay, now we'll just quickly recap a little bit more and then we'll again come to the questions. Uh, yes. Yeah. So we were studying about the host. So in the module 1, we have studied about the different kinds of host that we can use. We can have prokaryotic host in the and eukaryotic hosts. And then we have studied that what is the metabolism of the different kinds of hosts. Okay. Host metabolism. And then we have studied that for the multiplication and growth of the host organism, what kind of medias, what kind of growth medium are needed, and what are the essential components of a growth medium that should be added. So there should be proteins because these are the building blocks, the energy sources such as carbohydrate, sugar, you need lipids, you need fatty acids, and then obviously the genome, which is DNA, RNA, and then your vitamins, minerals, basic thing that human body required, that all things are required in mammalian cell culture especially. Then we have studied about bacterial growth kinetics. So why this bacterial growth kinetics study is important? Because accordingly, uh, okay, why you think the study of bacterial growth kinetics is important? Can anybody tell me why this slide was included in this in context of genetic engineering and whatever we have studied up till now, why you think knowing about the bacterial growth kinetics is important? Anybody?
Because certain products are sec- um, produced during certain growth stages.
Like,
Ma'am, like antibiotics and others, such products are usually made during the stationary phase.
yeah, right. So basically, for example, we say secondary metabolites. So secondary metabolites are produced during stationary phases. So you have to understand. So when you are doing large scale production of any product, let's say some antibiotics or metabolites, whatever it is. So you need to have the knowledge of at which phase the product formation would be the most. So if I want to produce secondary metabolites, if I want to produce antibiotics, I cannot use the lag phase, can never be used. I cannot use the log phase. I have to use the stationary phase for the production of secondary metabolites. Okay. And during large scale production, so when you have bioreactors, okay, you have bioreactors, and you have different kind of culture batches. You have different kinds of culture ways. You have batch culture, you have fed batch culture. I don't know whether you have studied about it or not. And you have continuous culture. So in these kinds of culture, you need to have the knowledge of the kind of phase which is the most appropriate for the production of your product, because then only you can decide whether you have to use a batch culture or a fed batch culture or a continuous culture. Okay. So for example, if you want to have a product which comes out in exponential phase, then you should have a continuous culture. Okay. So the type of culture that you are going to establish in a long I mean in a very large scale, for that you need to have the knowledge about the stages of a normal oh, happened. Oh, sorry, I think there was some network problem. Am I audible? uh, Hello, am I audible?
Yes, ma'am.
There was some network issue, it seems. So just let me again share my screen. I'll just check it's recording. Yeah, fine. Um, just a minute. Okay, I hope it's recording. Anyways, so what I was telling was that so on large, when you're doing a large scale culture, there are different ways in which you can set up your culture. You can set up in a batch manner or a fed batch manner or a continuous manner. So let's say if your product formation occurs in the exponential phase, then you have to use a continuous culture setup. So the type of culture setup that you have to use in a large scale production, for that also you need to have the knowledge of the phase in which your product is forming. So therefore, we have studied about the different phases of a bacterial growth curve. There is a lag phase. So lag phase is the adaptation phase. So when your bacteria is going into a new media, so before it will grow exponentially, it will take some time to adapt to the new environment. Okay. So for the adaptation of new environment, this is your lag phase. Just a minute. I just have to check whether it is recording or not. Otherwise, I hope it's recording. Anyways, yeah. That is your lag phase, where the adaptation of the microorganisms to the new media takes place. Now depending upon the compatibility and how good your media is, how rich your media in the nutrients, and how fresh your media is, the time of lag phase will depend. If your media is very fresh, full of nutrients, then the lag phase time would be very less. If your media is not very good, it's very old media, it's not very compatible with your organism, then the time of lag phase would be more. Then comes your exponential phase, where the exponential growth of the bacteria takes place. That means your growth rate is greater than your death rate. Okay, so when scientifically I put that the growth is exponential, then I have to say that the growth rate is greater than the death rate. That is your exponential phase. Then comes your stationary phase. In the stationary phase, your growth rate is equal to the death rate. Okay, so there's a plateau, stationary phase. And then comes your phase, where your growth rate is less than your death rate. So that is how you define. So it was not there in the module, because it is not the part of your this thing syllabus, but just for your information per se. So this is a very very important topic for your gate examinations and for your dbt as well. But forget, it is very important. A lot many questions from bioreactors and your questions from the growth phases, numericals especially, are asked. So if you get any chance, you should always read about it. And there is a book, Pauline M. Doran. Okay, Doran. It is out of the syllabus. I'm just telling you for your own benefit. So this Pauline M. Doran is a very good book for this thing, bioprocess. In fact, it is the only book I think which is good for bioprocess engineering. So if you are thinking of giving GATE exam, you should definitely go through this book. Okay, coming back to the topic. So now we know the information about the host. Okay, what is the metabolism of the host, how the host reacts, what are the different kinds of host. Then we start with the basics of cloning, how the cloning has to be done. So very basic of cloning, what we have to do first. I want to clone a gene of interest, and my gene of interest is, let's say, X. So, what is the first thing that I have to do? First, I have to isolate this X, then only I can start my cloning. Now, I need your gene of interest. Gene of interest has to be first isolated, and your plasmid has to be selected. Okay, now I want to isolate this gene of interest. So, for the isolation of my gene of interest, I can use different approaches. My approach depends upon how much information I have about the gene of interest. Okay, if I know the gene sequence, and or if I don't know the gene sequence. If I know the gene sequence, it is very easy. I'll just set up a PCR, and then I will isolate that gene of interest. I will amplify. How I will do? I know the gene sequence. I know that this is my gene sequence. From in the whole genome, this is my gene sequence. So what I will do is that from the whole genome, I have in NCBI, in UCSC genome browser, I have the sequence. Okay. Suppose I am working on mouse. Okay. So mouse, basically there are MM10, like these are libraries. So from this library, I will design a primer which is specific for the particular gene of interest that I want to amplify. And then with the help of those primer, I will amplify my gene of interest through polymerase chain reaction, PCR. But if I do not know the gene sequence, then I can either use the genomic library or cDNA library. It's a tedious process, and I will screen it, and then I will see the clone which contains the fragment, and then I will clone it. It is a tedious process. Normally nowadays what happens, that suppose if I, let's say I want to clone a gene, stat5. Okay, let's say I want to clone this gene. So this is a gene which is involved in TGF. I don't know you know or not. There is a pathway which is known as TGF beta pathway. So this gene is involved in TGF beta pathway, which is involved in proliferation. And this gene is also mutated in many forms of cancers. Anyways, so now I want to this is my gene of interest, stat5, and this I want to clone in a plasmid, say puck19. Okay, just for example. Now, for cloning of my stat5 into puck19, what I will do is that I will go to NCBI or I will go to UCSC. Then I will download the sequence of stat5, and then I will design primers from it. Okay. But one thing that you need to very well understand, very basic of cloning is, I'll just make a diagram and I'll let you know that this is stat5 gene. Okay, this is the primer I have designed. both forward and reverse primers. And this is my puck 19 vector. Now I want to insert my stat 5 into this puck 19. So I have to digest it also. So that means that when you are designing a primer, you design a primer with the help of some online tools available. Okay, that is okay. You can use primer 3, and you can also design primers through NCBI also. But manually, I have to add the restriction enzyme sites here, and these restriction enzyme sites should be the ones that are present in puck 19. So before adding the sites, I will just go through puck 19 vector map, and I will see what are the sites available in puck 19. Suppose puck 19 has eco R1, it has zo1, it has bam h1, and in my lab, or like whatever, in whatever context, I selected eco R1 and zo1. I have to clone my fragment here. So here manually in the primer that I have designed, I will add eco R1 site here, and I will add zo1 site here. Then my primer is completely designed. Is it clear to everybody what I'm trying to say?
Yes.
Also, uh, for you, before the restriction enzyme site, you can also add leader sequences. So you have some famous leader sequences. I don't exactly remember the sequence, but that you can add before the site. So that helps in the proper digestion. It helps in a proper digestion. So this is how you design your primers. So now I have designed my primers. I can simply, with the help of PCR, I can amplify my gene of interest. Now my gene of interest is ready. What I have to do now? Okay, these are like the basics. If you do not have a gene sequence information, then you have to construct a genomic library. So for the construction of genomic library, what are the steps? You have to isolate the genomic DNA, then you have to restriction digest it, purify. So these are like the basic steps for creation of a genomic library, and the steps for creation of a cDNA library, and then basic screening, how you can screen it, then polymerase chain reaction. So polymerase chain reaction, very quickly, three steps: denaturation, annealing, and elongation. So denaturation is the step in which, so basic, you have your this thing, DNA. Now you want to amplify it. These DNA are through, with the help of hydrogen bond, it is a duplex DNA. So for the polymerase chain reaction, for what you have to do, first you have to separate the two strands. Okay, this is my first strand, this is my second strand. So I will denature the DNA in such a way that the two strands get separated, and for the separation of these two strands, I will provide a very high temperature so that these DNA strands can get separated. So therefore, denaturation occurs at approximately 95 to 98 degrees Celsius, depending upon your, I mean, you have to standardize it, but mostly 95 to 98 degrees Celsius. So now I have denatured it. Now, okay, my two separate strands are ready. What I have to do now? I have to anneal my primers now. Whatever primers I have to anneal that. Whatever primers that I have prepared, I have to anneal it. So now I will anneal the primers. Therefore, anneal means I will join the primers, and therefore it is known as annealing. The second step is annealing. So primers always add at the 3' end. Why you add primers at the 3' end? Because this is 5'. So obviously you have to add in 3', so that synthesis occurs in 5' to 3' direction. Okay, I have added my primers. Now what I have to do? Now I have to elongate it. So your third step is elongation. So primers will prime the reaction. It will initiate the reaction. And then, because in the PCR reaction you also provides DNTP externally, then depending upon the sequence, the complementary nucleotide will start getting adding up over here. And therefore your whole strand is formed. So from your two strands, you get 1, 2, 3, 4. Okay, so from two like this, you get a one and two molecules from one molecule. So this is how your PCR basic denaturation, annealing, and elongation occurs. Then we have studied about primer designing. So primer designing, there are certain criterias that you need to look when you are designing a primer. So that we have already studied, but I told you guys that if you really want to study about primer designing, you can come up and ask me questions, but nobody did, so that's fine. But otherwise, I could have shown you how to design a primer here only. But anyways, the basic criterias when you are designing a primer is the GC content, the melting temperature difference. The melting temperature difference between the forward primer and reverse primer should not be greater than 2 to 5 degrees Celsius. It depends upon, like, I take 2 degrees, some takes 5 degrees also. But then your GC content should be about 55 to 60%. And there should not be repeats, as in, if your primer has repeats like AT, or ATA, then TAA, TTT. So if like these kinds of sequences are there, then it should be avoided. Then your primer should not form self-complementary structures. That if this is my primer and this region is complementary to this region, so it can form loops. So this loop formation should not occur when you're doing a primer designing. So these are like the basic steps, basic things that you need to remember. This is like polymerase chain reaction basic steps: denaturation, annealing, and elongation. And what are the reagents needed? You need the DNA, you need primer, you need DNTPs, and of course, you need the enzymes. Then you have to analyze your PCR reaction results. So for that, you need this marker is basically ladder. So this ladder is basically commercially available in the market, and this ladder helps uh, to determine the size of the band. So suppose I ran a 1 kb ladder. Okay, I ran a 1 kb ladder. So in 1 kb ladder, it will suppose like this the ladder is, and suppose here the band represents 3 kb, and I want my PCR amplification will give me a band of, let's say, about 2.8 kb. So if this is a 3 kb band and my band is coming something like here, this is 2 kb, this is 3 kb, it is somewhere near the 3 kb line, then I can say that okay, it is approximately of 2.8 kb, so my amplification is right. So like this, you have to have a ladder, so you can have a comparison, and then you can see whether the PCR reaction has worked right or not. Okay, then we'll just solve 2 or 3 questions, and again then we'll move to the revisions. So as I said, the questions have nothing to do with the topic that we are studying, we are revising, because the questions are randomly made, and so that you can just have a basic idea of how your real exam would look like. Okay. The buffer pH of stacking gel in SDS pages. Stacking gel. What is the pH of stacking gel in SDS page? We have already studied this. pH of stacking gel in SDS page.
6.8.
Yes. 6.8 is the pH, and for resolving gel,
8.8.
8.8. So what happens when your pH is increasing? What happens? What happens to glycine
Ma'am,
in the
in the
resolving gel?
running gel, the glycine becomes charged, and it moves ahead of the protein. And in the stacking gel, the glycine the protein is stacked between the glycine and the chloride ions.
Hmm. So basically, in your stacking gel, the chloride ions and glycines are present, and your protein is sandwiched, matlab stacked in between. So therefore, it is known as stacking gel. In resolving gel, what happens? In resolving gel, your pH is increased from 6.8 to 8.8. Now your pH is increased. So what happens? That the now the pi, that means the isoelectric point of glycine is, I don't remember, it is around something 6 something 6, approximately 6 something is the pi of the glycine. So what happens? The pH gets more than the pi, Therefore, glycine gets a negative charge. And when it imparts a negative charge, it starts moving forward faster. And therefore, now earlier there was chloride, protein, glycine. Now glycine will start moving forward, and then your resolution will take place. Anyways, in UV visible spectroscopy, the shift of absorption spectrum towards longer wavelength is known as what
Bathochromic
happens?
shift.
Pathochromic shift. Yes. Next question. This also we have dealt, I think. Yes. For C terminal amino acid analysis, the peptide needs to be treated with C terminal.
Carboxypeptidase.
Carboxypeptidase. So C terminal is the carboxyl group and N terminal is the amino group. Even if you do not know the answer, what looks the most likely answer from this is carboxypeptidase, which will peptidase. Ase means enzyme. Peptidase means it is cleaving peptide bonds. Carboxy, it will cleave the peptide bond near that carboxyl terminal. So from the name itself says it all. Okay. Frederick Griffith is associated with the discovery of DNA sequencing method, radium, PCR technique, transformation in bacteria.
Transformation in bacteria.
Transformation in bacteria. Okay. Next question is, which antibiotic is commonly added in mammalian cell culture media? Azithromycin, streptomycin, penicillin, and or both streptomycin and penicillin. Which antibiotic is added?
Ma'am, option D.
Yes. So both penicillin and streptomycin is added. So when you are doing any mammalian cell culture media preparation, so now we have a cocktail which is known as penstrep. Okay. So we need not to add individually penicillin and streptomycin. We have a cocktail which has penicillin also and streptomycin also that we add. Then question number 16.
Ma'am, why have we chosen? Why have we chosen only these two antibiotics?
No, see, you can have a wide range of antibiotics which can be used, but over the years with the help of various standardization process, so you need to have an antibiotic which performs the required function also, and which has like least effect, negative effect on your media also. So like whoever has standardized the use of penicillin and streptomycin, they have just stick to these and not the other variants of the same kind, because a, they are a little bit cheaper also, easily available, and b, because they do not have much negative, they do not impart much negative effect to the culture media. So that is why they are used, and they are also, like when you are adding your pen strap, you just have to add very little of it. Like if you have a vial, that vial can be used for a very long time, so it is economical also. So suppose if I am making a 50 ml media, so in that 50 ml media, I need to add only 50 microliters. So very less. So that is why pen strip is mostly used. Then next question is, some features are governed by the environmental behavior while others are not. Which library is used for understanding the phenotypic behavior governed by environment? cDNA library, transcriptome library, genomic library, both A and B, both A and C.
Ma'am, both A and B.
Both A and B. Okay, can you tell me the reason why both A and B?
Ma'am, in both we are we are actually dealing with the RNA.
Yeah.
So
So
that's
transcriptome library means we are studying about the transcriptome is the expressed region, and cDNA also represents the expressed region. And why we are looking at the expressed region and not in the genomic region? Because we are talking about the features which are governed by the environmental behavior. Okay. So how you think a feature can be reflected by the environmental behavior? For example, when you go out in sunlight, okay, so so when you are exposed to sunlight for a very long time, then your for a temporary or for a temporary basis, you you find that your skin gets tanned, and that tanning is because the overexpression or more expression of melanin which is present, melanin pigment which is present in the skin. That melanin pigment is expressed because of the environmental behavior, that is sun rays. And that melanin is very important because it protects our skin. That melanin also protects our skin from a lot of UV radiations and from a lot of cancers also. So that is why the exposure of the increased enhanced expression of melanin because of the environmental behavior, that is sunlight, that is a kind of a protective shield for us. So that is why so that this is one example where your some feature is getting expressed because of the environmental factor. So we always say a feature is getting expressed. So whenever we say that something is getting expressed, we always talk about transcriptome or cDNA library, because genomic library is nothing to do with the expression. Your expression has to be dealt with transcriptome or cDNA library. So the answer is A and B. D, yeah, both A and B. D. Okay, next question. And after these two questions, we'll again switch to some more revision. Okay. Raman was trying to clone the genome of a species X that has a genome size of about 300 MB. Okay. So he's trying to clone which has a genome size of 300 MB. Fine. He used bacteriophage as host for transformation. However, he was unable to generate the clone library. What can be the possible explanation for this? Bacteriophage did not have the receptors to take the ligated product. Bacteriophage was mutated. Bacteriophage carrying capacity was less than the size of the genome, or bacteriophage carrying capacity was more than the size of the genome. What do you think is the answer?
Ma'am, option C.
Bacteriophage carrying capacity was less than the size of the genome. So what is the carrying capacity of bacteriophage? I don't remember right now, but it is very less. So it is towards the initial side of the carrying capacity. So which has the most carrying capacity, that you should remember at least.
Yak, East.
Yes, yeast artificial chromosome, and we have back just before yak you have bacterial artificial chromosome. First is, I think, basic plasmids. Then I think there. Then I think after that only you have bacteriophage. Anyways, the answer is that it is 300 MB. Okay, the genome that he is trying to clone is 300 MB, and the carrying capacity, that means the maximum limit up till which a bacteriophage can clone a DNA fragment, is very less than 300 MB. Therefore, bacteriophage cannot be used as a host for this thing. So you have to understand, that is why we need to okay, just let it be. The question is not completely rightly framed, but anyways, the concept is that you should understand that depending upon the size of the clone, what kind of vector you have to use, and the type of vector not only depends upon the size of the clone, but also depends on and many other things. Nowadays you have so many different kinds of vectors which will provide you a lot of additional benefits. Like you have vectors which can be specifically used for CRISPR Cas technologies. CRISPR, like you have a vector which is known as PX458. Okay, this can be used for knock-in experiments, for CRISPR knock-in experiments. Then you have other vectors such as uh, PET 24X MS2 vectors, which are used for some other purposes. So now we are engineering vectors in such a way which not only provides us cloning and expression, but also provides us some additional advantages which can be used for some other experiments. For luciferase assay, that means we have studied about that Photinis spiralis, that is the firefly which is used in luciferase enzyme. Isolated luciferase is isolated from that firefly. So now we have vector which has a luciferase gene also, so that that vector will glow when you are doing luciferase. Or you have vectors which have JFP also in them. Okay, so suppose if I want to see that my DNA of interest has gone into the vector or not, I can see it through fluorescence also. Because if my gene of interest has gone, because my gene of interest has GFP, so it will fluoresce. So now the vectors are selected not only on the basis of the size and whether it is a cloning and expression, but also you have a wide variety to choose, so that you can choose depending upon your whatever you want to do, your experiment. Okay. Next question is match the following. SSBP is single stranded binding protein. So single stranded binding protein, what option should I choose from here? What it does? SSBP, single stranded binding protein,
Prevents reannealing of single strands.
prevents realigning of single strands. So, the name itself says that single stranded binding proteins will bind to the single strands. Okay, so from here, which option looks like the best choice? This one. So, why it will bind to single strands? So that the single strands will not realign and form a duplex. So that is why single strand binding proteins are needed. Then DNA ligase. What does DNA ligase do? It will have
Phosphodiester linkage.
phosphodiester linkages. So DNA ligase will, ligase means it is joining, and when you're talking about joining in the case of a nucleotide or DNA, that the joining is a phosphodiester bond. So DNA ligase will, because it's a ligase which is ligating DNA, therefore phosphodiester linkage. Then helicase will help in the unwinding of the two strands. C. Then tethering proteins, it will help in polymerase stabilization, and primase, it will help in the synthesis of RNA primers. So this we're talking about. These all enzymes are required in which process? In which process these whole enzymes are needed? We have
DNA
studied
replication.
DNA replication. So these are the enzymes of the DNA replication. Okay. So let's just quickly revise some more. Yeah. So we were talking about the gene of interest sorry, gene of interest isolation. And we'll try to finish this off. And if we are not able to finish the two lectures, so I think there would be 90%, I'm sure that there would be one more extra class next Tuesday from 7 to 9, the same time. And in that class, okay, what you want to do in the that would be the last final class. So how you want me to take the final class? Whether should I have n number of questions, or should I explain the concepts and questions, or you want to have some concept that I should explain to you, like whatever way you say, in which way you want, that would be best for your exams. Tell me. You
Ma'am,
want me
concepts and questions both.
concepts and questions both? Okay, another one was also saying something. Someone else was also saying something. You can also say. Mm. Concepts and question both. Is it fine for everybody, whoever is there in the class today? Concepts and questions. Okay, nobody's saying anything now. Anyways, so I'll take like this only. I will have concepts and questions both. And if you have any problem in between, you can always ask me. Like this only, I will try to have the questions and some questions. So the questions that we are doing today, some questions are the questions that I've already dealt, and some questions are little bit new. So the questions which are little bit new are important questions. I mean, the concepts are important, okay? Because those are the questions that have come in previous year assignments. So those are important ones. Anyways, so we were talking about the so we're talking about the basics of cloning. So we said that the first thing is that we want the gene of interest. Okay, we can isolate the gene of interest if we know the sequence, or if we don't know the sequence also. If we know the sequence, we can go through PCR. If we do not know the sequence, we can either through genomic library or transcriptome library, we can isolate it. That is fine. Then for setting up a PCR reaction, what are the what are the components we need, that we have discussed. How we can, so sorry, how we can analyze a PCR reaction, that we have dealt, that we need a ladder is basically artificially synthesized, available in the market commercially, which has the bands which corresponds to the particular size. So if I have a 1 kb ladder, I will have bands which corresponds to 1 kb, 2 kb, 3 kb, 4 kb, 5 kb, 6 kb, and then I can compare my band in comparison to this. Likewise, you have a 100 base pairs ladder also, and you have a 50 base pairs ladder also. Depending upon whether your product is very small or large, you can use whatever ladder you need. Then, what are the enzymes which are used in molecular cloning? So you need restriction enzyme, obviously, because you have to cut your DNA. Then you need polymerase for the amplification. You need alkaline phosphatase. Alkaline phosphatase removes the phosphate from the, which terminal alkaline phosphatase will remove phosphate? From which terminal alkaline, whether it removes phosphate from 5 prime end or from 3 prime end, alkaline phosphate.
5 prime.
5 prime end, because phosphate is present in 5 prime end only. At 3 prime end, no phosphate is there. There is OH. Now, alkaline phosphatase is important. Why? Because alkaline phosphatase will prevent the religation or realigning of your DNA, of your clone DNA. So, what happens? Suppose this is my vector, okay, and I cut it. Now, my vector is like this. Now, although I cut it with the help of restriction enzyme, here it is phosphate and here this is OH. So there are chances that this phosphate and OH can again come close together and they can again form the bond. But if I remove this phosphate, then because there is no phosphate, so there is no chance of again reformation of the bond between them. So that is why alkaline phosphatase is used to prevent the re-annealing of the digested product. But you have to also remember, so when you are doing digestion, not every time your alkaline phosphatase treatment is required, okay? Always whenever you are doing a cloning, always you have to use restriction enzyme, always you have to use ligase, always you have to use polymerase. But alkaline phosphatase not much required, depends whether you are getting religation or not. So you should remember that when you are doing a double digestion, okay, when you are doing double digestion means that you are using two restriction enzymes. This is your DNA, gene of interest. You are using a different restriction enzyme for this side, different restriction enzyme for this side. So when you are doing double digestion, then chances of religation are comparatively less. But when you are using a single digestion, then at that time the alkaline phosphatase treatment might be required. So that is why it is always preferred to have a double digestion, because double digestion confirms the orientation also, and also there are lesser chances of religation also. Anyways, so then we come to restriction methylase system. So you have to understand that I think this point is important because sir has also explained this point a lot when he was explaining, and in the this class also he took up the points. So one thing you remember, that this last lecture is very important. Why? Because whatever topics are covered in the last lecture, they are sure short very important. That is why they were included from the vast syllabus. Not every topic is uh, uh, dealt here, but these topics are very important. Like everything is important, but you have to pay extra attention to these topics also, plus the questions. And you can also go through the YouTube channel to revise it. I have to upload the last 2 or 3 videos. I will do that. So, okay. So this restriction methylase system, what does it mean? So basically, does anybody remember this restriction methylase system?
Ma'am, the restriction enzymes cannot cut the methylated DNA, basically.
So basically, the gist is that the restriction enzyme cannot cut the methylated DNA. Not always we can say this. This cannot be the definition, because we have dealt with DPN1. which can cut the methylated DNA also. So you have to understand that how we got to know, how we discovered the restriction enzymes. So we know that bacteriophage are the viruses that infects the bacteria. So whenever any foreign genome trying to, so bacteria has kind of made a mechanism to protect itself. So how it protects itself is that whenever a foreign genome, let's say the genome of a bacteria, sorry, bacteriophage is trying to infect the bacteria, it degrades its genome, it cuts its genome. So bacteria has some restriction enzymes, some enzymes that cuts the genome of the foreign entity. Suppose if a bacteriophage, if a virus is infecting a bacteria, so the bacteria will degrade the genome of the virus. But that bacteria restriction enzyme, if it is capable of degrading the genome of its virus, of the virus, then why it is not able to degrade its own DNA? Because bacteria knows how to differentiate between the DNA of its own and foreign DNA. How? Because the bacterial DNA is methylated, whereas the foreign DNA is not methylated. So therefore bacteria know how to restrict the entry of the foreign genome, and bacteria knows how to prevent its own genome with the help of methylase system. That is why it is known as restriction methylase system. Restriction of the foreign genome, methylase to protect its own genome. So its own genome is methylated, therefore it is protected, whereas the foreign genome is not methylated, therefore it is not protected. Now you can, I mean, this is for your examination point of view, it is not needed, but can you think, even I don't know, just now it came to my mind, that what if a virus kind of modifies itself in such a way that its genome is methylated. So if the genome of the virus is also methylated, then how bacteria can be able to differentiate between its genome and the viral genome? Then that genome is very difficult to degrade. I don't know whether there is such genome or not. That's just like one thing that came to my mind. Anyways, this is about restriction methylase system. You can see I just explained you the basics of what is the gist of restriction methylase system. And then you have different kinds of restriction enzymes. You have type 1 restriction enzyme, type 2, and type 3. So type 1 restriction enzyme, what will it do? See, the recognition, everybody will, every kind, the all three of these restriction enzyme will recognize, but whether they will cut at the recognition site or not, that differs. So type 1 will recognize the site, but it will cut 1000 base pairs away from the site. Type 3 will also recognize, but it will cut 25 to 20 base pairs away from the site. Whereas type 2, it will recognize the site also, and it will cut within the restriction site also. So if I ask that the restriction enzymes we use, like EcoR1, BAM, H1, ZO1, these are type 1, 2, or 3. So then what will you say? What would be the answer? These are what
Type
kind of
2.
type 2 restriction enzymes. So basically, when we are doing molecular cloning, we are using type 2 kind of restriction enzymes, not type 1, not type 3. And also you can see here that type 2 is producing sticky end also. um I'm not sure about this point, because we do have some enzymes that cut at the recognition sequence and they produce blunt end also. So I'm not very confirm about this point. Anyways, now we have the gene of interest. Okay, we have amplified it. Now what I have to do? Now I have to digest my gene of interest and my vector with the help of same restriction enzyme. Same set of restriction enzymes I have to use for the digestion of my gene of interest and for digestion of my vector. So we have dealt in detail how to set up this restriction reaction. So after your this thing, um amplification, next part that you have to do is this thing, digestion. So, and also one important thing, that when you are isolating your gene of interest, let's say you are isolating through PCR, that means you know the gene sequence. Mostly we know the gene sequence, mostly we do through PCR only. Then it's not just like that you design primers and then you set up a PCR reaction and then you got the product. No, you have to put a gradient PCR. Okay, gradient PCR means that amplification temperature, what should be your amplification temperature, you have to select, you have to take a range 60, 62, 64, 68, 66, and then you have to see at which temperature you are getting the brightest band and the most clear band, and that temperature you will select for further process. Okay, so you have to standardize the amplification protocol in such a way that you get the brightest band, and that standardization allows setting taking different temperatures, and what temperature you have to take depends upon the composition of your primers that you have chosen. That primers will decide what kind of temperature you have to take. Also the enzymes. Okay, that is okay, let's just leave it, that is far more detailed. Okay, so then when we put a when we have put a digestion reaction, then what we will do, your vector is digested, your gene of interest is digested. Now vector and gene of interest will combine with the help of ligase and you will have a recombinant DNA made. Okay, so that is the work of ligase. So ligase, it uses either ATP or NAD plus to catalyze the ligation reaction. Then you can, then alkaline phosphatase we have all dealt, it is used for removing of the 5 prime phosphate group, which helps in protecting the circularization or realigning of the uh, digested plasmid. Then we have linker molecules. So what are linker molecules? So suppose if your restriction enzyme is not able to create sticky ends, then you can have linker adapters or homopolymer tailing to generate sticky ends. That means if your restriction enzyme is producing blunt ends and not sticky ends, then those blunt ends can be converted to sticky ends with the help of linker molecules. Linker molecules also, adapters also, and homopolymer tailing also. Now we will come to vectors. So now, up till now, we have studied that okay, we have isolated the gene of interest, we have digested it. For digestion we have to use the same set of restriction enzyme, and then we will put a ligation reaction and then we will ligate it. Now after ligation you have to, after ligation you have to screen for the colonies, screen for your recombinant DNA. But before screening we will see what are the criterias of selecting a good vector. But first we'll just deal some few questions and then we'll come to that. Okay, okay, this is what we have just studied. The genome of the bacteria is not cleaved by its own restriction enzyme because restriction enzymes do not have the specificity for its own DNA. The DNA of the bacteria is non-methylated. The restriction enzymes sites are not present in the bacterial DNA, or the DNA of the bacteria is methylated. So what is the answer?
Option D.
Option
D.
D. The DNA of the bacteria is methylated. Okay. The next question is to analyze whether the column has separated the two molecules properly or not. What parameters must be checked? Distance between the two peaks, distance between the two peaks, average of base peak, resolution, or all of the above.
All of the above.
Okay, all of the, why you think all of the above and not just one of them?
Then because all those factors are necessary to make sure that the separation has been done properly.
Yeah,
If
so
with
basically,
only one,
yeah, yeah, continue.
I mean, if only one criteria is fulfilled, then uh, say if the distance between the two peaks is more, but the average base peak is base peak width is too much, then our separation has not been done properly.
Yeah. So basically, resolution, which determines whether the two molecules are separated properly or not, it depends upon the distance between the two peaks and average of the base peak width. Okay. So the resolution is directly proportional to the distance between the two peaks, and it is inversely proportional to the average of the base peak width. Matlab, if your peak is very broad, if you have very broad peaks like these, so that means the resolution is not proper. So there is also a term which is known as peak broadening, and if you have peak broadening, then you experimentally you try to perform so that you can standardize in such a way that your peak broadening is minimized. And then if you have peaks like this or like this, then there is these are not two separate peaks, the overlapping is happening, then also your resolution is not properly determined. So resolution depends upon these two things. Therefore, for analyzing whether the two molecules have separated properly or not, these two parameters are required. Then this question also we have dealt earlier also. So the difference between partition chromatography and adsorption chromatography is, in adsorption chromatography molecules are not adsorbed on the matrix. In partition chromatography molecules are not adsorbed on the matrix, whereas in adsorption molecules are adsorbed. In adsorption, matrix analyte forms mutual interaction that does not happen in partition. Both B and C. Which is the correct answer?
Ma'am, both B and C.
Yes, both B and C is the correct answer. Because so, okay, so we will anyways we will revise this portion also, if not in today's class, maybe in the next week class. But just for the uh, understanding of this particular question per se, so chromatography can be your partition or adsorption. So partition basically involves, if you have two your mobile phase and your stationary phase, if both are liquids, okay, then your analytes, it partition between the two phases. Suppose if the two liquids are water and oil. So one is an organic solvent, another is an inorganic solvent. And depending upon the property or the nature of your analyte, the analyte will separate between the two phases. So in liquid chromatography is a type of partition chromatography, where the analyte is partitioning between the phases. Whereas adsorption chromatography means that there is a column and there is a matrix, and in that matrix, sorry for such a bad drawing, very good at it. Yeah. So you have a column and you have matrix, and whatever your analyte is there, it is kind of adsorbing on the matrix. Adsorbing, so adsorbing means it is not going inside, absorbing means it is getting absorbed, it is getting inside. So when I put a ring on my finger, it is not absorbed, it is just present on my skin. But when I apply some ointment or some oil, it might get absorbed, it is not adsorbed. So that is your absorb and adsorbed. So adsorb means it is not going inside the matrix, it is just present on the outer layer of the matrix. So what is happening here in this kind of chromatography, in adsorption chromatography, your analyte, it interacts with the matrix and it is present on the matrix. It is not going inside, it is present on the matrix. So depending upon the kind of analyte and the kind of matrix, so some molecule, so if this is your analyte, it has A, B, C, and if A has the interaction capability with the matrix, so A will interact with matrix and B and C would be eluted. That is your adsorption. Now this interaction, it can be of any type. It can be your hydrophobic interaction, it can be your ion interaction chromatography. So this the kind of interaction that the matrix, it can be affinity chromatography, bioaffinity chromatography. So the kind of interaction that occurs of the between the analyte and matrix, that that can differ, but the basic principle is that in adsorption chromatography, molecules are adsorbed and there is some mutual interaction that does not happen in partition chromatography. In partition chromatography, just there is no interaction. Just depending upon the nature of the analyte, it gets partition between the two liquids. So you have to remember that liquid chromatography is a kind of partition chromatography, whereas your where sorry yeah sorry whereas let's say ion exchange chromatography is a kind of adsorption chromatography. Okay, now we'll just see one or two concepts again and then we'll solve the questions. So we were dealing about the good vector. So what is the basic need of vector? Because that vector will carry the foreign DNA and it is able to replicate in the host. Therefore origin of replication is very much required. And nowadays we have vectors which are known as shuttle vectors. So what are shuttle vectors? Anybody remembers what are shuttle vector? Anybody who remember what is a shuttle vector? So shuttle means what? You have shuttle buses which will take you from one place to another and again take you from the same place to another. Kind of a to and from motion it does. So from it takes you to both the places. Shuttle buses, if you have shuttle buses in your college, then it will take you from your department to the main gate and then again from the main gate to the department. It is taking you both the places. So therefore shuttle means it is shuttling between both the things. It has both the things. Therefore shuttle vector means that it has origin of replication of both prokaryotes and eukaryotes also. So such vectors can be used for prokaryotic function, matlab in prokaryotic host also and in eukaryotic host also. So basically that is why I was telling you that if you use a eukaryotic vector and you try to use a host of prokaryote, then you cannot replicate it. That is why in the first place when I asked the question that why you need the information whether your host is prokaryote or eukaryote, because when you have a vector which is eukaryote, then your host cannot be prokaryote. Your host has to be eukaryote. Then um yeah, so shuttle vectors are the vectors which have origin of replication for both prokaryotes and eukaryotes. So this is basically vector, the basic responsibility of a vector. Then what are the criteria of a good vector? It should be low molecular weight. Why it should be low molecular weight? Because if it is low molecular weight, then only if it is of a low molecular weight, it is easier for it to replicate. Okay, if it is of a high weight, high molecular weight, it is difficult for it to replicate. Then post entry into the host should give phenotypic changes. That means it should have some selection marker genes. Because if you do not have any selection marker genes, then how can you determine whether the vector has entered the host or not? Then there should be multiple cloning sites. What is the meaning of multiple cloning sites? So nowadays when you get a vector, so what kind of vectors are there nowadays? Suppose if this is a vector, commercially available vectors have MCS, multiple cloning sites. That means initially when the vectors were made, they just had cloning sites for one or two genes, EcoR1, ZO1, BAMH1, SMA1, just one or two restriction sites. But now nowadays, for the sake of convenience for researchers, vectors are made in such a way that you will have a region which is known as multiple cloning site, MCS region, where you will have restriction sites for n number of enzymes. That means EcoR1, BAMH1, ZO1, SMA1, you have so many restriction enzyme sites and you can choose any of them depending upon your convenience. So you have more options to choose to select the restriction enzyme site. That is known as multiple cloning site. That means, why? Because cloning will happen here only, na? So you need to understand that cloning happens between the restriction sites only. So now you have multiple cloning sites. If you have a vector which has eco R1 and zo1 only, so you are designing, you will design, when you will cut it at eco R1 and zo1, then your DNA will come over here. So that means this restriction sites are the sites where cloning actually occur. But if you have n number of sites, that means you have multiple sites for cloning. Therefore, that is known as multiple cloning sites. So multiple cloning site means sites where n number of restriction enzyme sites are present. Then high copy number. What is the meaning of high copy number? So vector can be of two types. Okay, the vector copy number can either be a low copy number plasmid or low copy number vector, or it can be a high copy number vector. So low copy number vector it means that when it is going inside the host, it will not produce many copies. It will produce like say uh, 5 or 6 copies, not many copies it will produce. But high copy vector means that when it will go inside the host, it is going to replicate more. It will produce say 200 or 300 or 500 copies depending upon the vector. So therefore we should try to have a vector which is a high copy vector, because high copy number vector will give us more colonies to screen, whereas low copy vector will give us very less, fewer colonies to screen. But again, for some really sophisticated and high end research, this cannot be the only criteria. And if you have a vector which is of a very low copy number plasmid, then you have to uh, then you have to put a plate which is known as vector control plate. Okay, what does it mean? So suppose if you have no option and you have to use a vector which is a low copy number vector, and you are getting just 5 colonies for example. Now 5 colonies can occur randomly also. Matlab, even if your transformation is not fine, then also there are chances that 2 or 3 colonies might occur. So how can we say that the colonies that are present over here are the colonies which are coming from transformation, or they are just noise, background noise? So for that we put a negative control plate. Okay, negative control plate which is known as vector control. So that means in this plate you are going to transform with only vector. That means these vectors which does not have your DNA, whereas these are the recombinant vectors which have your gene of interest. These are the cloned vectors, and these are simply vectors. So your, if your vector control plate is clear, that means no colonies are there in your vector control plate, and there are 5 colonies in your main plate, then you can say that the colonies that are occurring, that is because of transformation, and these are not noise. Is it clear to everybody what I'm trying to say?
Yes, ma'am.
So that means you have to understand that when you have a low copy number plasmid, then you have to have a vector control plate. Vector control plate is needed even in high copy number when you're doing it for the first time. But when you're doing a low copy number, every time you have to have a vector control plate. So always remember, whenever you are doing any experiments in biotechnology, or not just in biotechnology, any experiment in general, you have to have controls. Okay, you have to have positive control and negative control both. Positive control which will give result in each and every situation, anyways it will give the result, and negative control it will not give the results. So positive, what is the function of positive control? Suppose if you are doing an experiment and you are not getting result in positive control also, that means there is some problem in your protocol, or your reagents are not working, because it should give result in positive control always. So that is why positive control and negative control both are important when you are doing any experiment, whether it's a real time experiment, whether it's immunofluorescence experiments, whether it is Western blotting experiments, any kind of experiments needs controls. Okay, so then we have studied about the criterias of good vectors. Then what are the components of a vector? Vector should have an origin of replication. It should have marker genes. It should have promoter. It should promoter. If you want to have an expression vectors, you can have promoters. Then what are the different kinds of vectors which are available? Vectors can be your bacteria based vector, your phage based vectors, your yeast based vectors, your mammalian based vector, mammalian system based vectors. So these are the different kinds of vectors which are available. Then oh, sorry. Okay, we'll just do the questions now, 2 or 3 questions, and the rest of the part we will, like, whatever the we have kind of completed the first half, and then the whatever remaining part which is left, I mean the part which is yet to revise, that we will do it in the next class. So next class would be the last class. Hopefully next class is there. I have requested it should be there. And also again we are going to deal some more questions in the next class, different from these certainly, and that would be the end. So okay, first we'll do these questions. So match the following. Yeah, so hydrophobic interaction chromatography, what should be the answer here? Hydrophobic interaction chromatography.
Hydrophobicity.
Hydrophobicity, okay. So the basic uh, way by which hydrophobic interaction chromatography work, you should remember that how the water solvation layer, when you are putting salt, it is getting rid of, and how your hydrophobic area is getting exposed, that is causing the precipitation. So whenever you are, see, certain terms you should always, it should always come to your mind, it should be on your fingertips. When I am saying hydrophobic interaction chromatography, certain concepts like salting in, salting out, immediately it should come into your mind. When I'm saying affinity chromatography, then some nickel NTA column. So these are like some classic examples, nickel NTA column. And then so these things should come immediately in your mind when you say certain terms. When I talking about biotechnology applications, like we dealt in the last class, immediately it should come that when you're talking about applications of biotechnology for, let's say, in plants, bt cotton, classic example. So everywhere you will get bt cotton. So there are so many applications of biotechnology, so many recent applications coming, but certain examples are classics, so they are only dealt. That is why you are this disease resistant, herbicide resistant. So these are all classic examples. Then when you come to this thing, animal, in animal you talk about the therapeutics may that collagen factor 8 and factor 9, that is very important, very classic example. Then what else we have dealt, that animal breeding, that artificial insemination, embryo transfer, again very important, vaccines, monoclonal antibody production. So these are like these are hot topics, they will always come in any examination. If this is in your syllabus, questions from these will always will come. And I think I saw your assignment also, and in the last years also, I think you should try to focus on the part where they are asking that what would be the band size and how many number of bands, if you are cleaving your if you are running your gel, matlab protein on a gel, and you observe this number of bands, how many number of bands are there. I think that portion is also important, a little bit tricky, that is why that needs some extra attention to be put on. uh, okay, then in the last class we'll discuss some important everything is important, but I'll just try to analyze your assignment, and then we can just have like a quick revision of the topics, that hot topics that are asked in the assignments, and in general also they are important. Anyways, yeah, so ion exchange chromatography, it depends upon the
Charge
charge
distribution.
distribution. So depending upon whether you want to purify a cation or an anion, In that context, you use a cation exchange chromatography or an anion exchange chromatography. Cation exchange chromatography, the matrix is negatively charged. Anion exchange chromatography, the matrix is positively charged. Always remember it is the opposite. Then gel filtration chromatography, it is the surface area C, and then your affinity chromatography is the different ability to bind to certain molecules. It is D. Oh, I think I have already told you the answer of the next question. Anyways, so in cation exchange chromatography, the matrix has positively charged functional groups, negative charged functional group, neutral functional groups, and both. So what is the answer?
Negatively charged functional groups.
Negatively charged functional groups. This is the answer. In detail, we'll deal in next class. Okay. Next question is that also we have done. The first step in sequencing of protein is cleavage of the proteins with trypsin, with CNPR, reduction of the disulfide bonds, or none of the above.
Reducing the disulfide linkages.
Yes, reducing the disulfide bonds, because when you are doing the sequencing, if disulfide bonds are present, then it will interfere in the sequencing process. Therefore, disulfide bonds have to be reduced. It has to be treated. It can be done either with the help of oxidation by performic acid or reduction by DDT, diethiothitol. Then your next question is functional group identification can be studied by which of the
IR
following is
radiations.
infrared radiations. Yes. So this you should always remember that whenever we are dealing with this spectroscopy, absorption and emission, so we have the electromagnetic spectrum and we have different uh, uh, like different rays, infrared rays, UV rays, visible rays, and so on. So depending upon the energy that is available from a particular kind of rays, that is depending upon the energy that is available, the kind of application depends on that. For example, when you're talking about x-rays, so x-rays will give you a lot of energy, highest energy it will give you. So x-rays, can you use x-rays for the identification of functional group? Can you use x-rays?
No.
Why you cannot use x-rays?
Man, they will affect the bonds. They might break the bonds because they are so high
Because
energy wave.
yes, so because the energy of the x-rays will just take uh, the electron away from the atom only. Okay, so when I'm talking about UV visible spectroscopy, so UV visible spectroscopy has that much of energy that will take the electron from a higher energy level to a, sorry, from a lower energy level to a higher energy level. That much energy it has, but it does not have that much energy that it can just knock out the electron from the atom. It does not have that much of energy, but x-rays have though that much of energy. It is very powerful. So that is why the kind of energy that a particular wavelength is supplying, that energy can be used for that particular kind of study. Vibrational energies are not that much. Okay, spin energies are not that much. So for studying your vibrational energy, vibrational frequency, you cannot use UV rays or X-rays, because that will give energy more than that required for vibrations. So that is one thing you should always remember. The next question is identify the correct statement relating to the embryo transfer method. Okay. Eggs are fertilized and transferred to superior females. Eggs are fertilized in low breed females and then transferred to high breed females. Fertilization occur in vitro and the fertilized egg is subsequently transferred to superior female. Eggs are fertilized in the superior female and then transferred to low breed females. We have just dealt in, I think, last week this question. So, what is the answer of this question? What happens in
Ma'am,
embryo
option
transfer?
B. Eggs are
Option,
fertilized in the superior female and then transferred to low breed female.
yeah. So, first you have to fertilize in superior female, and then for the further growth, it can be, matlab, superior females need not to be uh, kept for the whole the whole development of the therefore, that at that time you need not to have superior females, you can have low breed females also. Then, what is the function of aminopectin in hybridoma technology? It blocks the de novo pathway, it blocks the salvage pathway, it blocks nucleotide synthesis, or all of the above.
Mom,
So
it
there are,
blocks nucleotide through through the de novo path.
yeah, it blocks the de novo pathway. So, aminopectin blocks the de novo pathway, but still salvage pathway is available, which can be used by the B cells. But myeloma cells, they do not have the salvage pathway. Therefore, since myeloma cells do not have the salvage pathway and the de novo pathway is also blocked, therefore myeloma cells will die. Whereas B cells, which do have the salvage pathway, but they cannot live for a very long period of time, so eventually they will die. But the hybrid cells that have myeloma cells also and B cells also, they will live because they have the salvage pathway to use from the B cells, and the capability to live because of the myeloma cells. So that is how, when you have your hat medium hypoxanthine aminopectin thymidine. So this aminopectin allow you to select only the fused cells, that is hybridoma cells, and it will kill the only B cells or only myeloma cells, and only fused cells, which are the hybridoma cells, only those cells will survive, and rest two cells will be killed in the hat medium. Okay, so for the next class, because you have exams also. Yeah.
Ma'am, here option C is also right.
No, we cannot say option C is right, because if I am saying it blocks nucleotide synthesis pathway, it is like half correct, because now nucleotide synthesis can occur through de novo also, it can occur to salvage pathway also. So it is not specifying whether it is blocking the de novo or the salvage pathway, because de novo salvage pathway it cannot uh, 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 block. But see, that is how you have to select. Here you have one specific answer. Suppose if you have a question that what is the function of aminopectin, it provides us nutrients, or second option is that suppose first option, it provides us nutrients, second option is that it blocks the nucleotide synthesis pathway, third option is that it is required for the proper growth of B cells, and D is, let's say, all of the above. Then in that case, you can select the nucleotide synthesis. It blocks nucleotide synthesis because all other options are entirely different. But here you have an option which is even more specific than this, which is the de novo pathway. Therefore, you will select this. So it depends upon the options that you have. Okay, this is here, though it is short different, but you might have options which looks very similar to each other, and you might think that this is also correct, this is also correct, so what should I choose? Then you should choose the option which is more specific and more related to the question. If you have a, like I said, that if you have options which are entirely different, like it provides us nutrition, it is required for growth, it is required for proliferation, and another option is that it blocks the nucleotide synthesis pathway, at that case you can select nucleotide synthesis. But here, because you have even more specific option than this, so you cannot select this. Okay, you have to select de novo pathway only. So yeah, so I was telling that in the next class, um two things we have to deal. First, that we will revise the energy charts for glycolysis and for Krebs cycle. Okay, that ATP production, that how many ATP, how many NADH, how many NAD plus are produced. I mean, how many FAD is produced, not NAD plus. How many FADH is produced? So that we have to see in glycolysis and in the Krebs cycle, which is important. You should have the knowledge, and that is not only important for your examination point of view, but that is important for, I mean, it is going to be there with you up till your uh, even in your PhD. That is important. So that we are going to discuss. Then we are going to discuss the rest of the topics which are left. We'll try to speed up a little so that we can complete each and everything. and then yeah, we'll deal some of the questions. And if you have some sorry Huh. And if you have some questions, some topics that you could not understand, you can ask me. So try to have some topics that are not, that you could not understand. So it would be easier for you, because then after last class, that is also an extended class. I mean, logically, today's class should be the last class, but you have one extended class next week just for your problem solving. After that, you won't have any class, and then you have your exams also. So okay, so from where we'll just end today's session. Uh, so read well. You have your exams and your, I mean, whatever your coursework also going on, but try to take some time for this course also and try to read. And whatever questions you have, we can discuss it in the next class. Okay. Okay, then fine. Bye bye. All the best.
Thank you, ma'am.